Hello students and welcome to this simple distillation lab presented by myself. My name is Hannah Yonts. I'm a third year biochemistry student at Brescia University. I'm also a student athlete on the women's volleyball team. Science has always been my passion and my favorite topic of learning, and that is why I'm going to continue to be a woman in science for the rest of my life. After I graduate from Brescia University, I plan on obtaining a doctorate of pharmacy. The reason why I chose this simple distillation lab is it was one of the most impactful and memorable laboratory experiments that I've completed so far. The reason why it was so memorable is I learned a lot during the lab. I was able to gain respect for science as an imperfect form of learning. For example, I made many mistakes during the simple distillation lab, however, I still gained a lot of knowledge. It's important to note that science is not about perfection, it's more about learning, and that's definitely a lesson that I was able to carry on with me throughout the rest of my studies. Enough about me, let's go ahead and dive right into the laboratory experiments today. Simple distillation is the process of heating and cooling liquids, collecting the water or the vapor from said liquids, and then separating the mixture. This is based off of differing boiling points. The goal of this lab would be to separate ethanol and water. Ethanol has a much lower boiling point at 78 degrees Celsius than water, and the reason why we would want to use distillation to separate them is it's otherwise very hard to separate. For example, this clear liquid, you cannot see the difference between the two liquids, so it's important that we use a scientific measure other than our eyes to be able to separate the two. In order to completely do this, the time estimate is about 4 to 6 hours. In order to complete this lab, you need an arrangement of all the equipment, which can be defined as an apparatus. The apparatus can be broken down into many different parts. First of all, I'm going to explain the heat source. Down here in this photo, you can see they're using a Bunsen burner, which is an open flame heat source. There's also a different variety of heat source you can use. My personal favorite and the one that I'm recommending that you all use is called a hot plate. It is about this big and it has knobs on the side. It's a flat surface that gets hot to the touch and it has no open flame. This is a little bit safer and it's also more accurate because you can use a small spin bar, which is pictured here to the side, that you would just drop into the mixture, which the spin bar is already in there for you. It is white. You would just drop it in and you would set the flask on top of the burner just as this one is set on top of the flame here. The spin bar spins and allows for the mixture to be evenly heated. That way, whenever the ethanol starts to boil, it can slowly rise into the West condenser and make for a more accurate separation. If you were to use a Bunsen burner, you would have to be very careful to be around the open flame, and also the mixture closer to the burner would be more warm or hotter than the mixture at the top. So it doesn't create for a very evenly heated mixture. It would still work, however. The most important part of this to keep an eye on during the lab, other than the open flame or the hot plate, is the thermometer. It's very important that you watch as the temperature rises closer to ethanol's boiling point, which we noted earlier that was 78 degrees Celsius. It's important to note the temperature because as your mixture starts to boil, the ethanol will slowly rise as the temperature gets closer to its boiling point, and the vapor will start to condense in this West condenser and flow all the way down to the collective glass. The West condenser is a two-tiered. It has an inside and an outside glass piece, and it allows for water to flow through the hoses on each side that connect to the piece. The water basically cools the ethanol vapor that rises up into the tube, and it allows for it to flow nicely into your collective glass. Lab grease can also be used to make the glass pieces, for example, right here and right here, fit a little bit nicer together. Sometimes your lab glassware doesn't fit necessarily evenly, and lab grease can just allow that to happen a little bit easier. For example, if I were to use lab grease on this flask, I would just apply it around the outside and then fit the glass pieces together. So that's basically the apparatus, or like I said, the setup of equipment that you need for this lab. It's pretty simple as long as you're safe and secure. So the results of this lab are pretty easy to tell just with the human eye. As we noted earlier, the ethanol's boiling point is 78 degrees Celsius. The ethanol vapor will rise into the West condenser and it will travel down to the collective glass. You know that you are finished with this lab when you can see that from your original mixture of it is separated about half and half, with half of it being ethanol in the collective glass and half of it being in the original glass, and that will be the water part of it. Once the sample stops boiling, the liquids have been separated, and that makes sense because water's not going to boil at 78 degrees Celsius, so that's when you know that all your ethanol has been removed from the water. In summary, the goal is just to complete this lab as accurately and efficiently as possible by separating the ethanol and water using their differing boiling points and proper laboratory technique. It's very important just to follow along and set up your apparatus and heat your mixture and continue to monitor your thermometer, all the steps we talked about, and just use scientific reasoning as your guide to complete this lab. Don't be afraid to ask questions or make mistakes. Make sure you're paying attention and wearing all of your proper equipment and being safe around the lab. And just remember that science is a process and it's about the imperfections sometimes, and it's okay to make mistakes as long as you're learning. That's definitely something that I took away from this lab the first time that I did it, and I hope you all enjoy it as much as I did. Thank you all so much for listening.